Welcome, everyone, and thank you for joining this Brain Inflammation Collaborative webinar today. I'm Christy Jagfeld, one of the co-founders, and I've been a patient advocate since 2012 when my daughter was diagnosed with POTS, EDS, mast cell, and PANDAS. She was part of a research study that was life-changing for our family, and I wanted to help to bring that to other families by finding more collaborative ways of participating in research. At the Brain Inflammation Collaborative, we know how frustrating it is to manage a chronic illness, and it's even harder when health tools aren't in your primary language. So we're excited to share our role in providing a Spanish-speaking version of our patient and research platform, where users can easily track symptoms, connect wearables, take control of their health journey in the language they're most comfortable with. This includes both of our websites as well. This is a huge step in our mission to reach underserved communities who are too often left out of clinical research, and we want to make sure our data actually reflects the real world, and every new voice helps us build a more accurate and inclusive picture of brain inflammation and chronic illness. We have a great webinar today where we'll hear from Dr. Hector Bonilla as he shares research on perspectives of long COVID, including its impacts worldwide and in the US. And the Brain Inflammation Collaborative's own Kelsey Aguirre will tell you about our free Spanish platform, patient platform, as well as a little bit more about how it works and why we launched it. And so I'd like to formally introduce you to our guest speakers today. We'll first hear from Dr. Hector Bonilla. He's a clinical professor of medicine at Stanford and the director of the Stanford Post-Acute COVID-19 Syndrome Clinic. As a young physician, Dr. Bonilla focused on the clinical management of HIV/AIDS and HCV, two neglected and stigmatized diseases for which effective therapies were in their infancy. In addition to teaching medical residents and students, Dr. Bonilla participated in numerous clinical trials and developed clinical research projects. Dr. Bonilla's interest in academia led him to the University of Pittsburgh Medical Center, where he was an assistant professor, clinician, and medical educator in the Department of Medicine in the Division of Infectious Diseases. Due to his interest in cytokines and immunological responses, Dr. Bonilla became a researcher at Immunoscience, a biotechnology company in California that works to develop a therapeutic HIV vaccine. Dr. Bonilla's experience of treating HIV/HCV, combined with his interest in inflammatory response is the driving force behind his desire to understand ME/CFS and long COVID. Next will be Kelsey Aguirre, one of BIC's data analysts. Kelsey earned her BS in biology from Texas Tech University, then moved to New York City and studied melanoma metastasis at Weill Cornell Medical College. Next slide, please. She continued to work in oncology as a junior scientist at a startup that focused on small molecule production. In 2021, Kelsey began her MS in biomedical data science at Icahn School of Medicine at Mount Sinai. She studied computational modeling under Vincenzo Fiore at the Center of Computational Psychiatry and completed her thesis optimizing a variety of models for behavioral data from severe OCD patients treated with deep brain stimulation. Thank you so much for joining us today, Dr. Bonilla and Kelsey, and I'll hand it over now to Dr. Bonilla.
Okay, can everybody see my slides? Okay, así ya no compré, assume yes. So now it's a pleasure to be here and it's an honor to be invited for this presentation. I'm going to talk about perspectives in long COVID and try to give kind of update on possible pathobiology mechanisms of this illness. So I have no a conflict of interest for this presentation. So I could talk about, I initially talk about the culprit of this problem of long COVID, the coronavirus, the human coronavirus. Most of the coronavirus are in animals, and for animals, they spill over humans, and for humans, cause human diseases. And they have 5 different genera of coronavirus. They call the alpha coronavirus, the beta coronavirus, the gamma, and the delta. The coronavirus associated with the pandemic are the coronavirus beta that include the SARS, the MERS, and the SARS-CoV-2, the one caused this pandemic and now cause all this kind of uh, condition we call long COVID disease. The coronavirus um, are RNA virus. It's a very long positive strain of RNA, and there are four major structural proteins: the spike protein, the envelope protein, the matrix, and the nucleocapsid proteins. And here in the bottom present like the spike protein because very important, important because it's the way how the virus attach to the ACE2 receptors in the human cells, and they have this kind of spike protein here represented in green. Is they call the binding domain. This domain, the changes in the domain make the virus more efficient. So the new variants of coronavirus it cause some mutations, and the mutation made the virus more infections and able to cause more disease compared with the prior variants. So how this happened? I think the coronavirus are the reservoir, the human coronavirus reservoirs in the bats for the SARS-CoV-1, SARS-CoV-2, and MERS. And from the bats, they can have an intermediary host. Then, in the case of SARS-CoV-1 and SARS-CoV-2, is the civet or small carnivores. In the case of MERS, is the camels or dromedaries. And those kind of if the people who handle those animals, they can get infected, spread to human, and cause a human disease, especially in people that are immunocompromised. Having reported the three major pandemic was coronavirus, was reported the first one in 2002, 2004 is the SARS. The WHO report around 8,000 cases, but one mortality around 10% of the people who got infected that die for this kind of conditions. In 2012, in the Middle East and Arabic countries, we have the MERS, and they report around 2,500 cases, confirmed cases, with mortality of 35%. And now we know at the end of 2019, beginning of 2020, we had the SARS-CoV-2 that have been infected more than the 100 million people have been infected, and many people had more than one infections with a mortality over 7 million people died as a consequence of this virus. So we know the SARS-CoV infections, or like I mentioned before, over 7 million people have been infected worldwide, many with a multiple infections. The around 5 million people have been death for this kind of condition, more men than women. It's interesting because in acute infection, the males getting sicker, go to ER, go to ICU, and the highest mortality in compared with the females. And the females, they have more the chronic illness, the long COVID, more of the main, like represent a difference in the immune responses in the males and females. So we know, for example, in females, response to vaccine. They respond better than the males to the vaccines and respond better to acute infection, but they are to develop this chronic illness. The same thing like we see people that get the shingles vaccines. The females are decrease the risk of developing dementia and compared with the females. So it's difference in immune responses. We know the mortality, as I mentioned before, higher in men, especially elderly population, in adults more than in children. And the people who die, majority of the people, adult people die is in the hospital, around 60%, around 50% die in the nursing homes. The comorbidity associated with the mortality in these patients include diabetes, obesity, cardiovascular diseases. And there are some groups, although not consistent, have been shown more affected by this kind of acute infection that include American Indians, Hispanics, and Blacks. But fortunately, majority of the people survive this kind of condition. And this study was published very early in the pandemic, from April 2020 to August 2021. Was a observation study from the UK, and they follow up 26,122 patients with acute SARS-CoV infections. In this kind of a figure, we see in blue are people who got COVID test positive. In green are the people who have a COVID test negative with respiratory symptoms, and they follow them from 5 weeks to 19 weeks. And every time they see the patients, they ask them for 12 symptoms in the last past 7 days. So when they evaluate, they found in green the people who have a COVID test negative. By the 12 weeks, almost everybody recovered. In contrast, the people who have a SARS-CoV infection, by the 5 weeks, around 12% of the patients had still persistent symptoms, but the 12 weeks, uh, over 3 weeks, reached the plateau. So maybe this is kind of findings is the one to help us try to understand or define this kind of condition in based or duration of the illness. So the question is, what happened with the people getting exposed to the virus? The beginning, the virus infect the respiratory cells because they have more the H2 receptors. If you see, H2 receptors are present almost in every single organ in the body, but the highest concentrations can be in the respiratory tract. As soon they infect the cells, they can cause acute injury, and the injury could end up with people with a pneumonia, ARDS, go in the vein, and when they recover, people end up having a pulmonary fibrosis. And sometimes this kind of fibrosis in the lungs can lead into the need of a lung transplant in some patients. They can affect the kidneys, cause acute renal failure, or a small family neuropathy, myocarditis, evidence of deep venous vein thrombosis, or pulmonary embolism. But it's another population that recover from the acute phase, but have a persistent symptoms. But there are people who have the ongoing biological dysregulation. When I talk about biological dysregulation, that possible evidence of virus persistent immune dysregulation, gut dysbiosis, mitochondrial dysfunction, or microvascular clotting. And from this kind of population, there are some people who have some symptoms. And the objective symptoms in this population could be people who present with PATS, postural orthostatic tachycardia syndrome, means when the patient go from flat to standard, the heart rate increased by over 30 beats, and this kind of increase in the heart rate has been persistent. Or people have inappropriate tachycardia, people few steps and the heart rate go from 60 to 100, or people develop orthostatic hypotension or hypertension, sleep disorders like a short sleep apnea, gastrointestinal disorders, diarrhea, constipation, nausea, vomiting, bloating. And there's another population that the people have no uh, clinical findings, only symptoms. People present with a chronic fatigue syndrome or present with fatigue, the post-exertional malaise, the brain fog, headaches, and other conditions. In addition, there's another people, like a long term, they increase the risk to develop diabetes, hypertension, autoimmune conditions, Parkinsonism, cardiovascular disease, aging, psychosis, and other conditions. So the spectrum of this kind of viral infection is huge, from the acute phases to the subacute or chronic phases to the increase the risk to develop other comorbid conditions. So what we know, long COVID in the United States have been estimated like around 6.4% of the adult population, 1.5% of the pediatric population experience long COVID symptoms. Mostly the females have been affected more than males, and around 20% of the people with a long COVID symptom, they have a significant impacted by this illness. Uh, this kind of COVID pandemic underscore the inequity, disparity happen in this country. Like we see the people with a lower income suffer more of consequence of this illness compared with people high income, and the cost of the care for patients from to the society range between to $6 billion, maybe more. The more time we have, more patients getting infected. So the healthcare cost from those patients increase exponentially. But this kind of long COVID, we consider part of the something we call infections associated chronic conditions that include other viruses like SARS-CoV-1, the MERS, the influenza, the mono, the chingle virus, Coxsackie, dengue, the chikungunya, measles, Ebola virus, Coxsackie, Borrelia, and Giardia. People after those infections, they can develop similar symptoms, and we believe this kind of long COVID is one of those post-infections associated conditions. So by definition have been kind of evolved from the beginning to the more recent definition is published in 2024. They call the nascent definition. Then this kind of definition define long COVID as an infection associated chronic conditions that occur after SARS-CoV-2 infections and is present for at least 3 months as a continuing, relaxed or remitting or progressive disease. Then the symptoms can represent one or multiple organ systems. So we see at the beginning of the here, start to the left, we have the acute infection, and the acute infection could present like a severe symptom, sometimes asymptomatic infections, and they can evolve to develop these kind of long COVID symptoms. So if we have a mild disease, do not exclude that you're going to develop these infections. Sometimes people develop these kind of infections after second or third infections. If the first one doesn't cause problem, it's a chance the second or third infection can lead into this kind of problem. Have been shown the more infection you have, the highest chance to develop this kind of conditions. And when the patient develops this kind of condition, have multiple symptoms. The most common symptom that we see in this patient is the post-exertional malaise. It's a symptom that patients after physical activity, stress, overstimulation, all the symptoms increase, maybe new symptoms appear during this kind of time. And this kind of post-exertional malaise have a delay in presentation. Patient do the activity today, maybe 1 or 2 days later could be worse. The fatigue, the difficult to for concentration, memory, process information, people call brain fog, or people have lightheadedness, or other people has PADS, sleep disturbances, and other symptoms. And the pathobiologic explanations about this kind of condition have been proposed multiple things that we can discuss later on, but patients present with a kind of multiple symptoms, not a single symptoms. For example, they can present with a cardiovascular manifestation that could be from arrhythmias, maybe clots, pulmonary embolism, DVT, or problem with the brain cognition affected, the memory, concentration, or migraine. In some cases, can be leading to strokes or mood disorders, or other people present with a PATS, postural orthostatic tachycardia syndrome, or other people have a MCFS or chronic fatigue syndrome. Other people present like kind of for autoimmune conditions like lupus, Sjogren, fibromyalgia, and other mixed connective tissue disorders. But remember, this kind of condition can affect anyone, can affect from the pediatric population to adult population, people with a low income, high income, sex, age, sexual orientation, race, ethnicity, location. Doesn't they are everybody can be subjected to be affected by the virus and develop long COVID symptoms. Majority of the people recover from this kind of conditions. It's a fraction that can change from some report like a 1.5 to around 10% of the population who have long COVID develop long COVID. The population who have acute COVID develop long COVID symptoms. The diagnosis is based on the clinical grounds because we don't have no clinical accepted biomarker for these kind of conditions. And remember, this kind of condition could be affect the people's functions and not able to work, to go back to the schools, were not able to have any functionality. And remember, those symptoms had been perceived for longer than 3 months to be considered as a long COVID, according to the nascent definition. So when you look at the symptoms, the symptoms can affect the population as a whole, and with a significant impact in the function, social, and economical consequences. And they are reporting over 200 symptoms, but some symptoms are more frequently than other ones. This study we did here in Stanford in our clinic, looking for 29 symptoms and looking the frequency and severity of those kind of symptoms. We have here represented in the X the different kind of symptoms from fatigue, post-exertional malaise, brain fog, and reflection sleep. And we have in the Ys, we have the frequency and severity of the symptoms. We have in in black is going to be the more severe, and in light gray is milder. So we see the most common symptoms. We see the fatigue, the post-exertional malaise, the brain fog and reflection sleep, headache, anxiety, lightheadedness, and the high frequency is correlation with the severity of this illness. So uh, how to explain those things have been proposed different pathobiology and based on some report or cases with some findings. For example, here, the fibrinoid macroclad complex here in the left upper quadrant. Then this is this study from the South Africa, and they found out in the plasma of those patients, there are some proteins, and they call amyloid. And this have been shown any virus can trigger expression of this kind of protein, and they stain green by using a special dye they call thiofluorin 4. And this kind of amyloid uh, proteins could be the matrix to develop making activations of the immune system and macroclad, and maybe decrease the blood flow in microcirculations, or people present with a dysbiosis or changes in the immune system, in the innate as well as in the specific CD4, CD8, or a or proteomics or autoimmunity, or people have mitochondrial dysfunction, and other people have some persistent of virus or reactivation of the virus. They can explain the symptoms. Let me go more details about this kind of pathobiology. So the group from South Africa, they look at the plasma of people who had long COVID, the spinning down, and from the pellet, they do those stain for amyloid, and they found this kind of proteins. So virus can induce this kind of inflammatory proteins. The neutrophils, they make produce something like a kind of net of proteins they call a, nets or netosis for neutrophils. And this could be one how to trap many virus particles or proteins as a way how we can protect it. Or sometimes there are some lipid components of this kind of complex. So here the acute virus, after exposure to fibrinogen, they can cause some changes in the structure of the virus, and the formation of this kind of amyloid. There are two pathways. One where the body is able to digest this kind of proteins, and the people will be able to recover. Or there's another group, they have some difficult to digest this kind of proteins, and they are develop this kind of microclads, and this could be causing this kind of problem. And here we have in the bottom in green, this is the stain for amyloid proteins. So the other things have been kind of interesting is the autoantibody. This is a kind of recent paper just came out, but it's important when we exposed to any viral infections, the immune system start getting activated, and we have a polyclonal B cell activations. You know, the B cells are the cells to make antibodies. So it's a diverse or production of antibodies that is different during the acute phase compared with the convalescent phase. And this activation is because of the molecular mimicry. So some sequences of the protein for the virus can activate a specific B cell, or could be a bystander activation, or epitopes, or the virus can spread it and they can activate B cells, try to make different antibodies. And this kind of in long COVID, they found out because of this kind of autoimmunity or production of antibodies and linked with autoimmune conditions like sarcoidosis, rheumatoid arthritis, and diabetes type 1. But the study has been very inconsistent, trying to see these antibodies persistent or not. So in this same study, they found out possible to transfer the IgG from patient with long COVID into a mice can recapitulate many of the clinical phenotypes that we see in our population. So the importance of this study because there are two groups very close to in time, but separated. One group was from Netherlands, another group here from the United States, Yale. They decided they have similar findings. So they got two populations. We have healthy controls and people have a long COVID with neurological manifestations. So they got the plasma from those kind of patients, and they found out different kind of proteins or antibodies, and they separate the antibodies and they inject into the mouse. And they found out the mouse start complaining, and the mouse has some kind of pain, and the mouse start reacting different. And when they're looking for this kind of antibodies, they found out accumulations in the sensorial neurons in the epidermis. And both studies show the similar findings. So this specify possible the autoimmunity could be have an important role in some of manifestation that we see in people with a long COVID. The other thing is the immune dysregulation by study of proteins or proteomics. So they got the plasma patients and they uh, analyze the different kind of proteins. Sometimes they analyze hundreds of those proteins, but they are this group they separate in different kind of categories of those proteins. mRNA expression that represent immune cells or antiviral genes, or other proteins than they are marker of apoptosis or cell death, other proteins to activate kinases, other proteins that have to do with the mitochondria function, other have to do with the regulatory or mitophagy, and other a protein that express high in this kind of population. So when they do this kind of analysis, and this study, they have 100 individuals and who have confirmed by lab they have a acute SARS-CoV-2, and they have a follow-up for 10 months after the infections, and they get the whole plasma from those patients and they look at this protein analysis. And they found out like three major groups, one group of proteins that represent immune activation, antiviral activity, like a protein like IL-6, IL-beta, IL-10, and others. Other proteins have to do with the mitochondria function, DRP1, the parking, and other proteins have to do with apoptotic markers, MCL1 and the LIVIN. And there are some proteins that are independent. They found the HIF1 alpha, IL-1 beta, IL-10, and NRF1. And so this one could be independent markers. So this kind of analysis of protein can explain the whole pathobiology process that we see in this kind of patient from inflammatory responses to mitochondrial dysfunction and cell death. But the one hypothesis has been like a kind of proposed and accepted for many people, like a very important hypothesis, the virus persistence. As you know, the virus, they need the ACE2 receptors, and the ACE2 receptors have been found out in many tissues. For example, here in this kind of figure into the right, we have in kind of in orange, the places have been confirmed this A2 receptor, different than the lungs is the most common things, but have been found the kidney, gastrointestinal tract, and unconfirmed in organs like the brain, nasal and oral cavities, and male reproductive system. And there are some viral particles have been detected in blood, in stool, and sputum. So it's something it's important to understand this kind of phenomena. And and the virus, when they are touched to the cells, the attachment lead into infect the cells, and later on the virus replicate. If you see any H2 receptors in many organs, for example, in endothelium have been shown to have H2 receptor, so we can explain the presence of black clots. There's some evidence in the brain, maybe explain many of the brain manifestations and brain inflammation that we see in those kind of patients. So it's important to understand the distribution of these eight receptors that can explain the multisystemic symptom that we see in this kind of populations. This study, they are looking for people who had acute COVID and they die. So they're looking for virus in different kind of compartments and organs. We see here in green, they are present. In orange, they are not present. And the other one maybe in no clear. But see, in the acute phase, the lung has the highest amount or infected infections particles, followed by the heart, the spleen, the liver, kidneys, bowel, and so have been found few viruses in brain tissue in those kind of those patients in acute phase. So what happened in people with a long COVID? So this study is a long-term follow-up. Here, I want to just mention, if it's white, means there's no virus particles, and the other ones in different colors represent the amount. With the dark blue, low concentration, dark red is highest concentration. We see in different kind of organs, from oropharynx, lower airway, lungs, we see a large concentration of virus particles that include RNA, subgenomic RNA, viral proteins, and even have been found out the presence of this kind of virus particles up to 203 days after the infections in many compartments have been found out in the esophagus, in the spleen, in the appendix, adrenal gland, ovaries, testes, and other things, another endometrium. And here in the right lower bottom, it comes out a stain for different kind of particles of the virus in different kind of tissues. Here in green, we see pieces of the virus in many organs. So in conclusion, we see in people with a long COVID, maybe the driver of this kind of inflammatory changes is related with the virus persistent in multiple organs, and this kind of epitopes, they want to trigger immune activation and inflammatory responses in this population. In the gastrointestinal tract, something have been proposed as a potential reservoir. Early in the pandemic, we have a study we did here in Stanford, and we have people with acute COVID, and we collect we almost weekly the PCR in the nose of those patients, and as well as we collect some stool samples, and we found out like in the stools, the virus persists longer than in the nasal tract, respiratory tract. So indicated like a possible the gut could be a reservoir for this kind of organisms. And people start doing some colonoscopy and biopsies, and they found all these kind of pieces of the virus or virus antigens or SARS-CoV-2 in the mucosa of those patients have been shown in the duodenum, in the ileum, and colon. So the other evidence of virus persistence has been looked at in plasma people months after they develop acute infections, and they found spike proteins in plasma in people without any evidence of acute infection before. So they think, oh, this kind of virus particles maybe is coming from virus hiding into a reservoir, and this is the one to activate the immune system, and this activation of the immune system bring of the consequence of different organs involved during the acute infections. For example, here the antigens, they're looking for antigens like a spike protein, the S1 protein is part of the spike protein, and nucleic acid proteins. And they found out around 25% of the whole population, they have a persistent virus particles in plasma, indicated possible is a viral reservoir as a driver of this kind of activations of the immune system. The other thing we're looking for is mitochondria function. This is a study when we did it in patients with a MSCFS or chronic fatigue syndrome and people with long COVID. We look at the lymphocytes, the T cells, the CD4, the CD8s, and the NK and B cells. And we're looking about the mitochondria function. And the way we look at mitochondria function is try to look at the production of something we call ROS, reactive oxygen species. It's a protein that mitochondria produce as a mechanism or response to infections. And when the production of this kind of protein is too high, they lead into more tissue damage and more inflammatory responses. So we found out in both in males and the females high elevation of this kind of proteins, but the females are higher concentrations. And the different kind of ROS change between the males and the females. In the females, we see the pathway of the superoxide dismutase. In the male, we see the lipoxidase. So indicate the impact of the virus in mitochondrial function is different in males compared with non-females. And here we can see the difference here. We have in blue is going to be the healthy controls. In red, people with MSCFS. And in green is people with long COVID who had MSCFS symptoms. And we see here in this kind of blood box, box plot in the healthy control is SC, have a lower amount ROS compared with in blue. In green is the people with a long COVID, and in red, the people with MSCFS. In the B cells, CD4 and CD8 cells. So it's something like we see this kind of changes in those cells imply like a mitochondrial dysfunction is going to be an possible and important pathway to explain many of the symptoms that happen in people long COVID. The other hypothesis is this biopsy. So the virus can change the oval microbiome. Remember, microbiome is the flora that live in our body, and the healthy microbiome is like a nature. The more diverse we have, the more microbes, different microbes, like we have more plants, more animals, clean environment is a healthy microbiome. In people with a long COVID and MSCF have been shown changes in this kind of microbiome to predominant of bacteria that are pro-inflammatory bacteria. Here, this study was done in 45 patients. They calculate stool samples from healthy controls and 45 patients with a long COVID. They matched by sex and age. And they found out in patients with a long COVID, they have enriched or the formiculates, and in the control, they found abundance in the phyla vertical microbiome. For example, here in the arrows, here in the right side, we have the genus, and we are looking for in people with a long COVID. We found in people long COVID, the Agrobacter and the Faecalibacter are higher concentrations compared with the healthy controls. And we have in the phylum, we see in the healthy control highest amount the vertical microbiota and as well as increase the acinobacteriota as a difference in the microbiome. And those kind of germs, can lead into more pro-inflammatory effects. So maybe they want to maintain the integrity of the gut as well as integrity of the mucosa. They are one of the proteins that bacteria they can produce called butyrate. And butyrate is very important to maintain the integrity of the desmosome in the cells and important to control inflammatory responses. The other things where we start working is try to study extracellular vesicles. Um, the extracellular vesicles is kind of interesting. Every single cells in the body eliminate these kind of little vesicles. And those vesicles initially they thought could be kind of way that do the body dispose of any kind of waste product. And they found out it's not. Those kind of vesicles could be a communicator with other cells, try to deliver a thing that but the other cells needed, maintain the integrity, They can deliver protein, they can deliver mitochondria. So it's like a kind of cargo proteins and try to maintain the communication between different kind of cells. So you can study from this kind of vesicles the how different are in size. From they are from a small to big size, or we can study the content of those vesicles. It can contain about the functionality of those kind of vesicles. And the other one, they are the source of this kind of vesicles. So we decided to study the source. And the reason we study the source is when the vesicle come out from the cells, they bring the cell membrane of the cells. So the marker of this kind of surface marker of this kind of vesicles can tell me about who is the parent cells. So we try to see what cells are activated or not activated in base of the analysis of these kind of proteins. And the thing we found out kind of very interesting. We found out like the here we have three groups: people who have long COVID who full recover. We have people who have long COVID who had MSCSS phenotype, and we have people with a chronic fatigue syndrome. There are people with symptoms before the pandemic appeared in our community. So we have in the first panel, we see the amount of those vesicles, and we found out in people with a long COVID and people with a MSCFS, the amount of vesicles are higher than the control, people who have complete recovery. The other thing we're looking for the B cells. In the B cells, we found out the amount of vesicles coming from the B cells are lower in patients with a long COVID MSCFS and patients with a MSCFS compared with the controls. And we have a higher concentration of those vesicles in MSCFS and long COVID, especially in the monocytes, platelets, and neuron cells. So this can tell me kind of few things. One, the population of cells have been activated in those kind of patients with long COVID, where the B cells is desired to make antibodies, can spread out to antibodies. The monocytes or macrophages activate in high amount. There are some cells to control inflammatory responses. The thromboinflammation, the brain inflammation could be related. We see the platelets elevated, and platelets are very important in try to clear infectious agents as well as in the thrombosis events that we can see in some patients with a long COVID, like DVTs or pulmonary embolism, and the neuronal cells that can explain possible the cognitive dysfunction that we can see in those patients. So this kind of analysis is can give you more information about the different kind of pathways that patients have. Maybe can help us to profile this kind of population. Maybe for therapeutics, you can say therapeutics try to block the different kind of pathway that every patient has. So, but it's important to know, let's go to practical things. Practical things is how we can protect each other, how we can minimize the risk factors. Here, I try to represent in green are factors to decrease the risk of the long COVID, and in red are factors to increase the risk of long COVID. For example, we see elderly population. Age is one of the factors that increase the risk of long COVID. So this group, the interventions, how to prevent to getting infections is important. Females are more prone to getting infections. Obesity, more prone to getting infections. Low income is one here associated with increase the risk of the long COVID, or people have multiple comorbidities. And there are some factors that to decrease the protect against the long COVID. Vaccination. Vaccination have been consistent study and shown vaccination decrease the risk of long COVID. And the risk to develop any potential complication with the vaccine is very, very minimal compared with the benefit we get with the vaccines. The early use of antivirals have been shown to decrease the risk factors. They having a healthy lifestyles, try to be in a good shape, try to prevent obesity. Those kind of things can decrease the risk factors. And the other thing, after acute infections, I tell my patients, wait maybe a couple of 2 or 3 months before you resume the normal activity. Don't push because when people do this kind of pushing after they recover from acute COVID, they may increase the risk of develop long COVID symptoms. The thing is how we can prevent long COVID. And the best way to prevent long COVID is try to prevent acute infections and prevent acute infection by having masks, good air quality, ventilation. And uh, when the people get COVID, we recommended if you recover, please rest it for maybe 2 or 3 months before you can reestablish normal activities. There is one study from Germany looking the astellin is a drug they use for allergies and they use in nasal spray and they found people on astellin decrease the risk of develop SARS-CoV-2 infections. And again, vaccination. This is a meta-analysis with over 500,000 participants and show 2 doses of the vaccine reduce the risk of long COVID by 36.9% and 3 doses by 67%. And the vaccination decreased the risk of cardiovascular events as well as atrial fibrillation. And the use of antivirals, the Paxlovid, molnupiravir, and have been shown some benefit in decreasing the risk of the very long COVID symptoms. And the post hoc analysis of metformin show the also decrease the risk of the very long COVID symptoms. There are some interventions maybe we can consider to use in our patients, try to prevent level of this illness. Treatments are going to be very briefly because there is nothing approved by the FDA to use for treatment of long COVID. But there are some interventions that we do as an off-label in our clinic. Is the patient is energy conservation activity, and patients one way how to control inflammation. The dietary changes, there are some diets able to modify inflammation, like a Mediterranean diet, and avoid sugars and carbs that have been promote increase of inflammatory cytokines. And we use off-label drugs in our clinic. The one we see benefit in some patients is the use of naltrexone and aripiprazole or Abilify.
Great. Thank you so much, Christy, and thank you, Dr. Bonilla. That was very interesting and informative, and I'm excited to hear the Q&A after. I'm just going to quickly run through some aspects of our Unahide platform and our newest rollout. So our Unahide platform is now available in Spanish. Next slide, please. What is Unhide? Unhide is a digital health platform that is designed to empower patients and caregivers by helping them track, understand, and share their health data. Through Unhide, patients can track symptoms along with health history in one place. They can connect wearable devices such as smartwatches, and they can generate symptom reports to easily share with their healthcare providers. Patients and caregivers can also receive paging notifications to help manage energy levels, contribute to research in 30+ chronic conditions related to brain inflammation, including ME/CFS and long COVID, and participants can elect to be contacted about clinical trials and research opportunities. Next slide, please. Okay, great. So here we have displayed a couple of examples of how the Unhide app looks on your phone. The first screenshot on the left is a view of how you can track your symptoms daily. The second is a monthly view of symptom tracking. So if you did a daily thing on the first picture and you zoomed out, that would be the kind of month view. And then on the right, we have a screenshot of how a few surveys display on the app. All of these functions on the Unhide application are optional and are designed to help the patient and caregiver navigate their symptoms. Next slide, please. You can also connect wearable devices. Here are 2 screenshots, and on the left, you can see how easy it is to link any wearable device you may already own with your Unhide platform. If you were to choose to link your wearable device, you would be able to see your past activity all within Unhide, just like the screenshot shows here on the right. Next slide. Here is another screenshot of how you can view your 30-day symptom tracker within your Unhide platform. If you choose to participate, you could even show your healthcare provider these easy follow-up reports of your recent symptoms in your next appointment. Next slide, please. Okay, great. Why launch Unhide in Spanish? Brain Inflammation Collaborative is committed to advancing patient-centered research that derives a better diagnosis and treatment. An important aspect of that is increasing patient accessibility. Physical challenges, energy limitations, and language constraints are only a few of the obstacles patients can encounter when seeking care. For many Spanish-speaking individuals, access barriers go beyond cost or geography. Language can affect whether someone participates in research, understands study materials, or even feels comfortable sharing their experiences. Historically, Spanish-speaking communities have often been underrepresented in research, and many patients are asked to participate in English. By providing access in Spanish, we can reduce these barriers, improve communication, and create opportunities for more representative and inclusive research. Next slide, please. So participating in Spanish. We've begun translating studies hosted on Unhide to make research participation more accessible for Spanish-speaking communities. This effort has been led by fluent Spanish-speaking members of our own team and currently includes studies such as the brain fog study, the what helped what doesn't, and the mood and behavior questionnaire. Importantly, these are not simple word-for-word translations. We are adapting the medical terminology, conditions names, and survey language to better reflect the cultural and healthcare context in which Spanish-speaking individuals receive their care. Our work also includes Spanish language versions of the standardized survey instruments such as the FS36 and FunCap 27. These are available on the platform as well. Expanding Spanish language research opportunities is an ongoing part of our mission to improve accessibility, representation, and equity in neuroinflammation research. Next slide, please. Here you can see the Unhide platform in Spanish. This is now available nationally for the Spanish-speaking community. And then the next slide, please. Unhide is an easy-to-use digital health platform accessible via our app designed to virtually bring patients, their healthcare providers, and researchers all together. You can scan this QR code to join now. Right now, we're especially looking to enroll children and teens, healthy controls of all ages, and Spanish speakers as well. We appreciate you sending the QR code or our website, unhidenow.org, to your network to help us drive our research. Now I will pass it back to Christy for our Q&A.
Thank you, Dr. Bonilla and Kelsey, for this presentation today. It's really important to keep pushing this work forward, sharing experiences, and highlighting the opportunities for research. So we do have some questions that I'm going to ask. See here. Dr. Bonilla, do you see a difference in the symptomology of patients who have had long COVID for many years compared to those who have long COVID more recently? Are there any trends in how this condition evolves over time?
They have been evolved because we have a different viruses, the virus and variants. The variants change. I think very early in the pandemic, we used to see a lot of people with a loss of sense of smells and taste. Now we very rarely see this kind of symptoms. So maybe the epitopes of the virus and changes could be changed in the symptomatology. Maybe the presentations or long COVID conditions going to change according to the variants. And there are some evidence possible the variants, so like a Omicron and pre-Omicron, has a different clinical manifestations.
Great, thank you. Do you think endothelial dysfunction can cause microclot formation in long COVID, or is it directly attributed to specific foreign antigens?
Endothelial dysfunction is one of the hypotheses that have been proposed in the long COVID. As you know, the endothelial cells, they have a S2 receptor, so they are potential, can be infected by the virus. So the virus can infect the cells and make the cells very dysfunctional. The second thing, there are many players here in the activations of platelets or activations of the monocytes. They are very important in the formation of the the thrombi or thromboinflammation, and possible they can contribute the formations of microclots, having shown a acute infection, the expression of proteins that change the 3D structure. They call misfolding proteins, and they are on sequences of those kind of proteins has on amyloid sequences. It's possible the virus changes and changes in protein expression can explain the whole kind of complex of thromboinflammation, immune system activation, and develop later on microclass or microthrombi.
Thank you. Do you think that EVs could one day be used as a biomarker for long COVID and ME/CFS?
Yes, this area I am working, I am kind of excited, and I think I think it is
Yeah,
because it's able to profile what kind of population of cells are activated. So in this kind of profiling, maybe you can try to see maybe combination of therapies, anti-inflammatory drugs, maybe drugs to control clot formation, or maybe antivirals. So this could be depends on the patient. We're going to have able to customize the patient treatment according to the AV profiling. But this study, we had 125 samples that we tested. I would like maybe to do in different populations with different kind of symptoms and see if any specific biomarkers for a specific symptom. By
yeah,
my feelings, yes, could be a good marker for long COVID ME/CFS as well as that profiling the patient symptoms too.
yeah. It's fun to hear the excitement of new studies and the possibilities there. So here's a question for Kelsey. Um, do you want to talk a little bit? Somebody had a question that was about finding the app in the App
Yeah.
Store. So actually, it's not called Unhide, but I'll
Called
let
My
you
Data
answer.
Helps. Yeah. Yeah. So it seems I was just following along with that as well in the Q&A. It seems when you search My Data Help within the iOS App Store, you find it easier. Also scanning the QR code on that last slide is a good way as well, or going to unhidenow.org gives you all the access as well to register for My Data Helps. But yes, it can be a little confusing with the My Data Helps and the Unhide name. Thank you, Serena.
Yep, there's the QR code. And you can also use a browser. You don't have to use the app. You do
Yes.
consent first, and then it does give you directions on finding the app after you've created an account.
Exactly. Thank you, Christy.
So, Dr. Bonilla, which possible treatments for long COVID would you like to see trialed or studied? You you did talk a little bit about some studies, and I guess the microbiome could be a piece in there as well.
Mhm.
Yeah, I think there. I think for me, part of the problem in those patients, or a big problem, is going to be immune system and inflammatory responses. Uh, Try to understand these inflammatory responses is very important because inflammation is good and is bad. Inflammation, if you survive, means the immune system activation is good. But if you have activation immune system and you die, is bad. So try to understand these mechanisms of inflammatory response is very important, and try to identify the specific marker of these kind of conditions, and you can target this kind of protein. So anti-inflammatory drugs and immunomodulators for me are the drugs have play a very important role in this kind of conditions. Um, Patients, they teach me a lot of things. The use of naltrexone, things I learned from patients, and I still learning from patients how to use naltrexone because still I don't know exactly how to use naltrexone. People think like it's easy, but no, it's more complicated than it is. But we see in some people very dramatic responses with naltrexone. And naltrexone, we know, is a anti-inflammatory drug and immunomodulator drug, has an impact in NK cell functions and had impact in cytokine inflammatory responses.
Yeah,
The
and that's one of the things with our study that the what helped, what didn't, in patients, and a lot of the symptoms overlap. We look at 30 plus different diagnoses, with long COVID being one of them, and the anti-inflammatories, how it does help with that inflammation piece for some people and different symptoms that can be helped with that. Do you think the autoantibody production is attributed to viral persistence, or can you get autoantibody creation independent of viral persistence?
I think they are, we don't know exactly the answer, but I think have been some purpose why the body produce antibodies. Is because one, you have the virus, and the virus some sequences. The way we make antibodies try to identify some sequences of the virus. So when they enter the sequences, they activate the immune system in a specific way. So possible for mimicry is one way. Possible virus persistent, antigen persistent, it could be the driver. And if the virus persists, the virus can maybe release some pieces of the virus. And these kind of pieces, they want to activate the B cells to make a specific autoantibodies. And those antibodies can recognize some our own body, like happen in lupus. Lupus is your body start making antibodies, but they recognize your body as an enemy. You destroy yourself. If one destroy yourself, you present all this kind of manifestation that we see in some patients. So the autoantibodies could be generated by virus persistent or persistent of viral epitopes, or maybe by mimicry, they can have a cross-reactivity and cause symptoms to us.
Thank you. What is driving netosis and etiosis contributing to the microclots? Are there any druggable targets to prevent netosis in humans?
So netopsis, I can't explain the neutrophils. We have a different kind of cells in the body, and one of they called the neutrophils. Like everybody knows the neutrophils, and the neutrophils they are way to proteins try to release this kind of proteins like a kind of nets. They are outside the cells, and they can trap. So it's a trap of different kind of epitopes, and this way they can control infection, control inflammation in this way. But sometimes when they trap protein that produce more inflammatory responses, and this can cause some damage, especially in acute SARS-CoV infection. The lungs have been shown in the pathobiology of acute SARS-CoV infection. The neutrophils has a very important role by this mechanism. But how we can regulate is very difficult because netopsis is not bad. Netopsis can be good, but when they go in the other direction, it's going to be a problem. How to have the appropriate volume or how to activate this kind of system is very difficult. But at least we understand a little bit more about the role of the neutrophils, especially in the acute phase or SARS-CoV infection, is very important.
Okay, we have time for one last question, and then I want you to also touch just really briefly at the end of the importance of Spanish-speaking participants to participate in research and the difficulties around that. But this is the last question. Recent papers show that there are two predominant types of long COVID. Long COVID 1 with microclots and activated platelets. Ticagrelor appears to be a good treatment candidate. Has it been considered? And long COVID-2, MCAS predominant. Has ketotifen been considered as a treatment?
I think many people try to confuse things more than for it's my own opinion. People try to confuse things and meant to clarify. They try to have classify type 1 and type 2. There's no a distinction, no black and white, these kind of things. This is kind of a spectrum. You have activations of the immune system here. And try to say type 1 and type 2. No, maybe you have to profile the patients. Maybe if this can help you to profile these people. But it's not like a clear distinction here. In the micro class is kind of important things to understand. This kind of class that have been reported as a secondary or activation or production of amyloid, they don't respond to anticoagulants very well. So it's a different kind of pathways of how to degrade this kind of accumulations of amyloid is difficult. The use and the regular drug that we use for platelets anticoagulation doesn't work in this kind of populations. So the area is more complicated. There are some studies looking the anticoagulants, like a combination of different kind of drugs, including Plavix and all those drugs, and the study have been not conclusive. And it's because of this kind of phenomena. The thing is very, very, very complicated. It's not as simple as we have. Understanding is very important. So the way to consider treatment for those people is keep on doing some research and try to make more clarity. The thing is not easy, um, and research takes time. I want to make emphasis. People come to my office, ask me questions, and I say, I don't have the answer for you. I'm sorry. The only way to get answer is try to get some data. And the only way you can get data is try to make the people to be take part of the studies. So
Exactly.
recruitment people for the study is difficult. I cannot manufacture data. I cannot bring this kind of put. I need to just bring the data, try to make sense. This reminds me very clear when one of the drugs I've been using in people with MSCFS and long COVID is aripiprazole. It's antipsychotic drug. How I found out these things? Because patients are like a guinea pigs. People are desperate and they go for different kind of treatment. And I remember this patient came to my clinic and she asked me to prescribe aripiprazole, antipsychotic drug in people with ME/CFS. I say, I'm sorry, ma'am, it's antipsychotic. I'm not going to prescribe for you. And I said no. And I called 3 months later and she told me she's doing better because she restarted taking the aripiprazole. So I pay attention to this kind of stuff and try to read about it. And she was taking a low doses. She's done no doses for depression, for bipolar disorders, schizophrenia. And she was very clear when I asked her to prescribe this medication. I don't have any mental illness, depression, but the only thing that worked for me. And I found out by patient experience and listen to the patient that this drug has anti-inflammatory properties, has able to regulate the immune system, and has a possible impact in protection of the neurons in the brain. But this is only data. We need to do clinical trials. We need to understand this kind of process better. But the listen to the patient is very important. But again, we cannot make the data. We need to have the data. So when I present to my boss, is I went to my boss and said, listen, I'm doing something in the clinic. Maybe here in Stanford, you can see like it's crazy. Me using antipsychotic treatment of patients with a with MSCFS. The response was, show me the data. And it
Yeah.
show the data and show some clinical benefit. So it's very important to give an answer to the patient, to show you the data. We need to have everybody participate in the trials.
Yeah.
Sometimes people have difficulty and being some excuses. They don't want to try anything, but people they do by themselves, something could be dangerous. There are many providers doing irresponsible things from those patients, promising drugs that have no benefit, and the people spend the little resources they have, income. I see people broke because different kind of treatment the cost for every treatment $2,000 or $3,000 out of pocket. For regular pharmacy, it's going to be a crime to do things for patients.
Yeah.
I think we need to be honest with our patients, tell the truth, don't make like a false promises because we don't have a magic pill or silver bullet now.
Yes, thank you, Dr. Bonilla. That is very true. And we are collecting data, and we hope at some point we could do a study with you. And people need to participate, or because, like you said, we can't make up data, and that's the only way that real change is going to come. So I thank both of you, Kelsey and Dr. Bonilla, and I want to close here with, if you're able to donate, please consider it. We're a 501(c)(3), and research is really very expensive, and the recruiting process and all of the pieces that go along with it. And a lot of times we like to try to help reduce barriers to the research itself. And we do need help in making this a sustainable project. So um, we would like you to consider joining us in our next webinar in July. It's going to be on pediatric long COVID featuring Dr. Rachel Gross. We look forward to engaging with everyone soon, and make sure to follow us on social media. We have a great social media team and a great scientific writer, and we have some great educational posts and current research updates. So thank you for attending.